Good morning. Hope you all are doing well. What I'm going to try to do this morning is talk through protein synthesis, hoping that maybe that will help a little bit. The central dogma simply says that DNA is going to be used to make a specific sequence of RNA, which will in turn code for specific proteins. Now, this is all dependent on complementary base pairing. And you know already that for DNA, adenine goes with thymine, thymine goes with adenine. For RNA, adenine goes with uracil, uracil goes with adenine. Cytosine goes with guanine, guanine goes with cytosine for both of them. Now, protein synthesis happens in two basic parts. The first part is called transcription, where we're going to use DNA to transcribe RNA. And this is going to occur in the nucleus of the cell, primarily so we can protect the DNA as it's used. Now, look at this sequence of DNA. You can see that it is formed as you would expect by complementary base pairing. But notice that in this particular process, unlike DNA replication, we're not going to use the entire DNA strand. We're only going to use a portion of it that we refer to as a gene. So we're only going to unzip the gene, not the whole strand of DNA, and we're going to use one side as a template or a pattern. And so that's what you see here. You see only the gene has been unzipped. And the way we do that is we use enzymes to break these hydrogen bonds that are right in here. And then we're going to use complementary base pairing to transcribe the gene and to create what we call pre-mRNA. So notice, complementary base pairing says G will go with C. Remember, we're creating a strand of RNA. G will go with C, T with A with U. Remember, in RNA, there is uracil, not thymine. A with U, G with C. And so this little strand right here is what we call pre-messenger RNA. And it's going to get released, and our DNA is going to reestablish those hydrogen bonds. It's going to rebind and then rewind. And so then this is what you'll end up with. Your DNA, and notice guys, it has not been changed from the original at all. And here is your pre-messenger RNA. And this is very interesting because we're going to have to do some processing, and we'll talk more about that in our lecture part. But for pre-messenger RNA nucleotides that are called introns, we're going to cut those out. And then the remaining segments, when the introns are removed, are going to be spliced together. And those segments that are going to be actually used are called exons. So the mature messenger RNA, once it's been processed, leaves the nucleus through the nuclear pores to the cytoplasm and finds ribosomes. Now, the second part of protein synthesis is what we call translation. And it's going to happen in the cytoplasm at the ribosome. And here's where we pick up another form of RNA. Ribosomes are made of ribosomal RNA and proteins. So we have our strand of messenger RNA, mature messenger RNA, that we had manufactured. And I wrote it in the other direction so you can more clearly see. It's divided into segments of 3 nucleotides. These are called codons. Codon 1, codon 2, codon 1000, however many we have. Now, the ribosome can read the messenger RNA one codon at a time. That it's going to use complementary base pairing to match that codon with a transfer RNA anticodon. It's also carrying a specific amino acid. So let's look at the structure of transfer RNA. Here on one end of it is your anticodon. Here's another transfer RNA. There's your anticodon. Then there's a representation of your transfer RNA. And then, of course, there's your specific amino acid sitting on top. So notice what's going to happen. Your CCA codon is going to call for an anticodon that complementary base pairs, which would be GGU. GGU just happens to be carrying a specific amino acid. And then the ribosome moves over to the second codon, and it reads UUC, which calls for an anticodon of AAG. And that brings your second amino acid into place. And when they're in close proximity, the amino acids will peptide bond with one another. So this is a peptide bond right here. And eventually this forms a protein. Go back to YouTube and watch the video from DNA to protein 3D, and that might be helpful. I'm also going to do another drawing for you. Okay, so let's suppose that you and I are starting with a strand of DNA. Figure out how to hold this, okay? So we're starting with the strand of DNA, and I'm just going to make this up. C, G, T, A, T, T, G, G, C, C. So we know the other strand of DNA would be G, C, A, T, A, A, C, C, G, G. Okay? And that's all complementary base pairing. So notice I used my top strand to make the matching complementary strand of DNA. Now remember, the next thing I have to do, I have to use transcription to make messenger RNA. So what I'm going to do is I'm going to say, okay, well, let me use this second strand of DNA and get the complementary base pairing to give me my messenger RNA. So G will go with C, C with G, A can't go with T, it has to go with U, T will go with A with U, A with U, C with G, C with G, G with C, and G with C. So remember then, and mine's not going to work out correctly because I didn't think about it, but you can see now that I can divide that messenger RNA into codons. Okay? So this is a very, very important part. Now you want to know what are going to be my transfer RNA anticodons. And you must get those from the codons, of course. So C will go with G, G with C, U with A. That's my first anticodon. Excuse me. A will go with U, U with A, U with A. That's my second. G will go with C, G will go with C, C will go with G. That's my third one. And then, of course, G would start the beginning of the next one, whatever it happened to be. Okay, so now I have my transfer RNA anticodons, and I want to use my code to be able to look up what amino acids they belong to. And guys, this is very important. To look up your amino acid, you must, and this is where a lot of people make mistakes, you must use your messenger RNA. Do not make a mistake and use your transfer RNA. Okay, so I just happen to have that genetic code, and our first one with our messenger RNA is CGU. So let's look at this. Notice that the first base will be listed in these 4 rows, okay? So my first base is a C. So that puts me in this row. Notice all of these start with C. Okay. My second letter is a G. So you see up here it says, what's your second base? Is it a U, a C, a A, or a G? Well, mine's a G. But remember, my first one was a C. So I stay on the C row, follow down until I see CG. Notice all four of these are CG. Now, my third letter was a U. So, my third base, I already know I'm right in here, and they say, okay, well, if you're a U, you are CGU, and you correspond to the amino acid arginine. So, then when you are writing down your amino acids, you only have to write the code, okay? And so the first one is ARG. All right, notice my next one is AUU. Okie dokie. So I find my first base is an A, which puts me in this row. I find my second base, which is a U, puts me in this column, this row. AUU is ILE. So I write that down next. Okay. My third one is GGC. So again, I go G, G, C, gly, and I write down gly. And I continue that process until I have completed my strand of amino acids, which will give me my protein. So I hope this helps, and if you have questions, please don't hesitate to let me know. Take care, guys. Keep working hard.